Welcome to this story seminar series. Before I introduce the speaker today, so first I can acknowledge his Story family, actually Ian Story, his wife, and his son here. So there's a little bit of story. This is actually a very special year since Professor Story arrived in UC Davis as a professor in zoology 100 years ago. And this is a seminar series. It's the most prestigious of seminar on campus. So since 1960, And then without the very generous of the support from the Story family, there is no way we can invite, esteemed scientists like Dr. Spencer to come here to give us such a wonderful talk. So we will have the public seminar today, and then tomorrow we're at the same place. We'll have more of a scientific specific talk. And then, so first I'd like to introduce Dr. Tom Spencer. So he has many titles. So he's creators of the distinguished professor, from the University of Missouri, Columbia. And then he also is a vice chancellor of the research and scientific director of Next Gen Precision Health, founding director of the Interdisciplinary of Reproduction and Healthy Group. He received numerous of awards, related to the reproductive biology, developmental biology. I just named a couple of them. So received the Physiology and Neurology Award from American Society of Animal Science, a research award, a distinguished fellow in Society for the Study of the Reproduction. And then Dr. Spencer also elected as a member of National Academy of Science in 2019. So his research is primarily focused on reproductive and developing biology, particularly function of the uterus and placenta. And then he's very, very productive, published more than 270 peer-reviewed papers. It's very, very impressive, which H index of 120, which is very, very, very high. So we're glad, Professor Spencer will come to UC Davis to share, this great talk with us. Without further ado.
I really appreciate that kind introduction, Wei Jun. Actually, so I first met Wei Jun. Actually, both assistant professors at Texas A&M together, and we had our labs basically on the same floor. And so I got to actually become both colleagues as well as good friends with him. And last year, he received the NIH Prize in Food and Agriculture, and so that's a great accomplishment. I think I take a little bit of part in that because at Texas A&M, he said, I have this opportunity to go to UC Davis, and I said, you should definitely go. And so I'm very proud of him, and it's really an honor to be here. And this year's recipient of the NIH Prize in Food and Agriculture is also from UC Davis. So it's quite an honor to be here at a place that has really a storied place in terms of research and a commitment to the land-grant university concept. So today I want to talk about dual purpose with dual benefit, kind of give you an overview of some of the research programs that I've been involved in that kind of highlight how research in domestic animals can benefit both human as well as animal health. And then I'll end up on some topics that I think are pretty important and emergent in terms of the reproductive biology field and what we can do with some of the newer genetic technologies in domestic animals. So first of all, I have to give a hearty thanks to the people that really make research fun. So this is my lab group. I have a joint lab group with a very talented assistant professor named Andrew Kelleher, who's shown on the right in the beard. I have some great former lab members and some great colleagues and collaborators, both within Mizzou as well as in the U.S. and in Europe. And I've been very fortunate to have been funded by the NIH and USDA, as well as a number of other entities, for a number of years to support our research endeavors. So the research overview, I've been involved in really looking at development and function of the uterus, initially using sheep, and now we transitioned to both using mice as well as human tissues. The basic mechanisms of the establishment of pregnancy, again, I used to use sheep. Now I've gone to cattle, mice, as well as I do directly work with human patients with some clinical colleagues. And then the genetics of fertility, again, we started out in beef, now we're involved predominantly in dairy cattle, and we use the power of mouse genetics in a lot of our different research programs. So without a doubt, domestic animals have had a tremendous impact on our knowledge of human health and the development of biomedical applications that improve the human condition. Sheep have been a premier model for maternal fetal medicine, and really a lot of our advances in neonatology as well as pediatrics is in a large part due to studies in the sheep. With pigs, again, a primary impact in terms of neonatal matology. Basically all the formula that is used for babies in the U.S. and worldwide were tested and first formulated and tested in pigs. And they are a great translational biomedical research model that we'll discuss at the end of the talk today. Horses have revolutionized what we know about reproductive immunology, where they actually have a portion of their placenta that's rejected by the mother and that they have a perfectly normal pregnancy. And then in cattle, basically all the modern day assisted reproductive technology procedures, such as IVF, IVM, ICSI, and embryo transfer were all originally developed and pioneered in cattle. So animals have an essential role in terms of helping us improve the human condition. We also must remember that they also have an essential role in human health via nutrition, both the provision of meat and milk and high protein and fat sources for the human diet. I want to highlight some of the work today that we call dual purpose with dual benefit. This is a publication that a group of us who held a meeting a number of years ago that was highlighting the successes of a joint program between the National Institutes of Health and the USDA National Institute of Food and Agriculture. So in 2010, a group of scientists successfully lobbied to create this program. This funded research with domestic animals that impact both animal agriculture as well as animal and human health. In over a 9 to 10 year period, they supported 61 grants totaling about $107 million. And importantly, almost half of the grants were actually allocated to either new or early stage investigators. So a lot of the people that are in the field today, particularly that are now associate professors, benefited greatly from this program. And what was really important is you could get NIH level of funding for animal agriculture projects and really do some things that were very unique. We were able to get this program renewed once, and then unfortunately the NIH decided to drop out, and there's not enough funds in the USDA to support the program. But many of us that collectively are shown here as authors of this paper really benefited greatly, and many of those grants also were awarded to scientists here at UC Davis. Now, the ones that predominantly I was involved in are focused on fertility that we can basically define as the pregnancy rate to a single insemination. And as you can see here, if you're in a multiple ovulation species like sheep or pigs, pregnancy rate is pretty high. But when you go to a monoovulatory condition like a horse or cattle or humans, fertility rates precipitously drop. And so we basically be engaged in trying to understand how do you improve pregnancy rates so you can have better production in terms of animal agriculture, and also understand fundamental mechanisms that you can use to improve pregnancy rates in women. So subfertility is a major problem in domestic animals, companion animals, as well as humans. And pregnancy loss really is a major limitation to production efficiency, but also the efficiency of assisted reproductive technologies. And for a lot of human patients, there's a significant investment, both psychologically as well as financially, in becoming pregnant through assisted reproduction, and anything we can do to improve their condition is very important in terms of an overall women's health research goal. So what determines pregnancy success? Well, it's both the father and the mother, the reproductive tract environment, the vagina, the oviduct, and to be honest, the uterus. We're going to talk a lot about the uterus today because I love the uterus, to be quite honest. I think everybody who's ever known me, I have a coffee cup that I drink coffee cup every day. I drink out of. It's Captain Uterus. It has a human uterus on it with the word captain on it. And so I've studied this organ for many, many years and find it fascinating. Another determinant is the developmental competence of the embryo. So the genetics of the father and mother as well, there are significant epigenetic influences as well. And overall, these combine to impact fertility and are also the origin of many pregnancy complications, such as preeclampsia and fetal growth retardation. And ultimately, the health of the offspring is also dictated by the success of pregnancy. So we're going to talk a little bit about cattle now, and I kind of want to orient everybody where you have fertilization in the oviduct or conception, and then the morula stage embryo enters the uterus and then undergoes a process where it forms what's called a blastocyst. And in cattle and other domestic ruminants, you undergo a marked elongation of the outer layer of the placenta called the trophoblast. And this is when the conceptus signals its presence to the mother to maintain progesterone production. Progesterone is necessary for pregnancy. And so within the uterus, you have two specialized regions. One is you have an intercaruncular region that has large numbers of glands. These glands produce substances that collectively are components of the milk of the uterus that bathe the early embryo. Then you have a caruncular area or caruncle that doesn't have glands, and this interdigitates with specialized areas of the placenta that forms the foundation of placentome for pregnancy. And so this placentation process in cattle is pretty amazing. You know, it takes roughly about 3 months to have a fully established placenta, and this includes both the development of a placentome and an interplacentome region. But what is really important is within the placentomes, you have the development of these unique binucleated cells. They have 2 nuclei, and they're a foundational cell type in the cotyledons and placentomes. They produce very unique hormones that are needed for pregnancy, like progesterone, placental lactogen, and pregnancy-associated glycoproteins. And they also stimulate maternal adaptations to pregnancy, so they stimulate mammogenesis to allow for provision of milk to the offspring. Now, we have traditionally studied the early stage of pregnancy, and the reason is that if you look at pregnancy loss in cattle, you'll note that the majority of pregnancy loss occurs during the first 2 months of gestation. This is very similar to what you see in humans as well. If you get through the first trimester, you actually have, and you establish pregnancy, the success of pregnancy and a live birth of a normally developed fetus and a healthy offspring happens. And so we really tried to focus on how to understand this pregnancy loss during the first 2 months and how to solve this from physiological and genetic perspectives. And so one area that we focused about 2 decades in is really looking at early pregnancy events, and particularly studying the process where the embryo has to go from about a 500 micron sphere to a almost 20 centimeter in length conceptus, which looks like a really thick piece of dental floss that's producing the pregnancy recognition hormone interferon tau and establishing presence to the mother. And the reason that we're looking at this period is because early on in my career, we developed the uterine gland knockout model in collaboration with Skip Bartol at Auburn University. And basically, we expose neonatal lambs to a non-metabolizable form of progesterone, and they normally would develop hundreds of glands, but in the uterine gland knockout, we have prevented gland development, and so the adults don't have glands, and they're infertile due to recurrent pregnancy loss. So this basically established the concept that the glands of the uterus were secreting factors that were important for early conceptive survival and development. And we went on to go through a series of studies over a number of years with very talented graduate students, which basically found that we have normal blastocysts in these animals, but they fail to undergo this conceptus elongation for pregnancy establishment. And so we spent many years actually trying to understand what are the factors from the endometrium, particularly the glands, that are secreted into the uterine lumen that drive this conceptus elongation and pregnancy establishment. And at the same time, we actually started to dabble in cattle. And the reason we did is because we established a great collaboration with Pat Lonergan and his group at the University of College Dublin. And they were studying very similar things, and so we both found together that the endometrium goes through a tremendous gene expression change event, which you can see up here, where between about day 7 and 13, in response to progesterone, you have a upregulation of large numbers of genes. And since progesterone is the progestational hormone, we sought to understand genes specifically that would be induced by progesterone in the endometrium. But the problem was we really couldn't discriminate, like if we study all these genes which are upregulated, like which ones are the critical ones. And some of that actually started to be highlighted in some work that actually Harris Lewin, who's in the audience, was involved with, and a number of other colleagues across the world, which were basically showing that the endometrium could sense the difference between an in vivo normal produced embryo and one produced by IVF or one produced by nuclear transfer. And so we actually started to dabble in this area because of that seminal work that was done by Harris Lewin's lab and others. And we chose to use kind of a novel system at the time, where I read this paper by Bill Macmillan in New Zealand, and he had reported that you could basically continually transfer in embryos into dairy heifers and segregate out animals that always got pregnant or rarely ever got pregnant. And so I thought that this would be a really great model where you could basically study the endometrium that was fully functional and one that for some reason was not as functional. And so basically we thought that this would be a good way to study innate differences in uterine competence to support pregnancy. And so we started this project that I'll highlight some of the findings. And we did this in collaboration with Tom Geary at the USDA ARS Fort Keogh in Montana. So what Tom and his colleagues did was took a group of heifers. So we took about 300 initially, and we put in a single in vitro produced embryo. That actual embryo was produced at the University of Florida by Pete Hansen's group and then flown to Montana where we did the embryo transfer. And so we basically looked by ultrasound in pregnancy diagnosis, and we asked the question, did they establish pregnancy or not? And then we cycled back the animals, and we did this over and over and over. And what we were able to do is to diverge out a group of animals that always got pregnant, some that were subfertile or only got pregnant 1 or 3 or 4 out of times, and then a group which was completely infertile and never got pregnant to a single embryo transfer event. And then we started to study their uterus. And kind of the penultimate experiment was we drove these animals from Montana down to, I was at the time, Washington State University, and we took 2 in vivo produced embryos, transferred them in on day 7, and then we collected them on day 17, which you normally would have this conceptus elongation event. And so we did 21 high fertile, 10 subfertile, and 5 infertile. And what we found is there's no real difference in pregnancy rates between the high fertile and the subfertile animals, but only 1 out of the 5 infertile animals got pregnant. And you can see the different types of conceptuses that we recovered, and we saw a tremendous range in the length of the conceptus in normal, high fertile animals, a little less elongation in the subfertile animals, but they were still elongating, and only 1 out of the 5 infertile animals had a conceptus that was actually completely spherical. The problem to this day, we still don't know if you're a long conceptus, do you actually establish pregnancy better than a short conceptus? And we never solved that problem, and it turns out, you want to talk about later why it's so difficult, and I'll be happy to entertain you. So the first thing we did is we looked at all the genes in the conceptus, and we found about 558 genes up in the good conceptuses from high fertile animals, about 729 down, but we found that literally most of the genes, about 19,000, were not different. And it turned out some of the genes that we identified that were lower expressed actually in a mouse system, if you delete those genes, actually they cause defects in embryogenesis or placental development, but it turns out that you have to delete both copies, so that probably isn't the reason why the conceptus was underdeveloped. And then we asked the question, again, using transcriptome analysis, could we take the biopsy of an open animal and essentially say, do they have a better chance of getting pregnant based on their response to an embryo? And so first we did open animals, and essentially you cannot discriminate the endometrium from these different fertility classified heifers based on their transcriptome. And it turns out, to be honest, you can also not do that in women. You can't, endometrial biopsies on the cycle before you do embryo transfer are completely not informative in terms of pregnancy outcome in women nor histology. But what we did find is if you looked at the pregnant versus the non-pregnant animals, in the high fertile animals, you can see a tremendous response to the endometrium. So you have about 3,422 genes that go up or down. But in the subfertile animals, you only have about a third of that. You have about 1,096. And so it looks like the ability of the endometrium to respond to the conceptus is diminished in the subfertile animals. So they have an inappropriate response to the conceptus. And it turned out that a couple of groups a few years later actually showed the same thing in biopsies of women undergoing IVF treatment, that you have a diminished response to both progesterone as well as the conceptus, and that tends to be associated with the development of preeclampsia and fetal growth retardation. So kind of in summary, cattle have innate differences in the uterine competence to support conceptus survival and development. You have important biological mechanisms that contribute to subfertility and infertility. We have reduction in conceptus elongation or growth. diminished response of the endometrium to the conceptus, with the majority of the pregnancies are lost by the first month. But what we found is that at least in heifers, that is animals that have never had a pregnancy before, you actually have a good uterus. So that means that there must be some differences in the conceptus that also contribute to pregnancy loss. And as a sidelight, we did parlay all this knowledge into, we currently have 2 different NIH grants where we're sampling human patients at Duke University and doing a complete workup on their endometrium, basically so we can develop diagnostic tools to help women understand how many IVF cycles that they have to go through before they might get pregnant, and also inform clinicians in terms of potential therapies that we can use to ameliorate pregnancy loss in them. So for the next couple of minutes, I just want to talk about a new project that we have, which are what are the sire contributions to pregnancy establishment. And so what we really think is that between the period of day 30 and day 60, actually the sire or the paternal genome really has a strong influence on whether or not you have successful pregnancy establishment. And so this is some work that we're doing in collaboration with Sophia Ortega at the University of Wisconsin and Kai Poehler at Texas A&M. And we switched to dairy cattle for this because there's about 1,200 active dairy sires in North America, and most of them have been genotyped and sequenced. And you also have individual sires that are mated to very large numbers of females, so you can get very informative pregnancy rate data on them when you have literally hundreds of thousands of matings on each sire. And their records are curated both by farms as well as national databases that are maintained by the USDA in Beltsville. And it turns out that actually there's more than 4 million dairy and beef cattle in the US that have been fully genotyped. And in Ireland alone, there's about 1.5 million. So really, records analysis can really help you out in terms of these types of questions. And really, our hypothesis was that the sire has a strong influence on pregnancy establishment. And so we used a mechanism called sire conception rate, and this is a value that you can obtain for all dairy sires in the United States. So you have a negative number, you tend to not establish pregnancy as well. If you have a positive number, you're better at pregnancy establishment. So we literally just took animals in this category and animals on the other end, and we started to phenotype them in terms of their ability to establish pregnancy upon natural breeding. We basically found, we basically found that fertilization rate was affected in these low pregnancy rate bulls, embryo production was lower, conceptus elongation was not compromised, but we got a reduction in heifer pregnancy rate in the low SCR bulls. And as you see, if we take a group of bulls that are either high or low, there's really no difference in pregnancy rate on day 19, not a huge difference, but by day 33, you have about a 30% reduction in pregnancy rate. And so what we've been trying to understand are what are the developmental processes in the embryo that are contributing to this. So is it a defect in embryogenesis, or now we know mostly it's a defect in placental development with a reduction in the development of these binucleate cells within the placenta. And this actually goes good with some work that was originally done by Kai Poehler, where he showed that the amount of these pregnancy-associated glycoproteins that are specifically produced by these binucleate cells are lower in animals that are experiencing pregnancy loss within the first month. And so they're a biomarker of pregnancy loss and thus pregnancy success. And so our current projects is we're continuing to phenotype and genotype sires. We're doing in vivo studies of pregnancy establishment. We also have an in vitro system where we're studying placental differentiation. And we've been doing a lot of single cell transcriptome analysis because basically what we know is that we don't know anything about placental development in cattle. Like we don't understand the different types of trophoblast, and we don't really understand how the placenta develops. And until we do that, we can't really understand the mechanisms of pregnancy loss when you have a defect in placental development. So we've done a lot of single cell analysis of the developing placenta. This highlights some work done by Kim Davenport on a mature day 195 placenta. You can just see that using single cell analysis, you can basically understand different cell types that are present in a complex tissue. And so we found varying levels of different types of trophoblast, as well as other known cell types within the placenta. And what's quite interesting is you can bioinformatically use this data to predict which cell is derived from which. So trajectory of how a uninucleate cell develops into a binucleate cell, and then we can study this using our in vitro stem cell, trophoblast stem cell assays by doing genetic engineering to knock the genes out. And I think one thing that was very surprising in this work is normally placental development is strongly influenced by transcription factors because they are primary regulators of cell differentiation. But what we really found was that when binucleate cells develop, they actually lose almost all of their transcription factors. And so we think this is a very unique developmental process, and it may require a lot of epigenetic or non-genetic means where you have epitranscriptomic regulation, and we're currently working in this area of looking at DNA methylation, non-coding RNAs, as well as RNA modifications. So I want to end talking about dual purpose and other impacts and maybe highlight a few things that are in today's modern press, as well as highlight some assets that we have at the University of Missouri. So I'd like to really highlight what we call Precision Health in Pigs, or the Big Pig program, where we're developing translational medicine models in pigs using genetic engineering, because traditional model species like mice, C. elegans, Drosophila don't have the same type of physiology that's needed to understand and also be a good model for human health. So wild pigs, they're monogastrics. They have human-type physiology and human-type organs. Most all modern-day medical devices are tested in pigs first before they're deemed safe for humans. They also are very useful for xenotransplantation. They have the full range of human weights. You can model chronic medical conditions like diabetes, cardiovascular disease, and cancer. You can also model acute medical conditions that you can't model in rodents, such as premature birth, trauma, as well as sepsis. And we're very fortunate to have an unbelievable team at Mizzou, Kristen Whitworth, Randy Prather, and Kevin Wells, that collectively run an NIH-funded National Swine Resource and Research Center, as well as we're the large animal testing component for the the somatic cell gene editing effort out of the NIH Office of the Director. And really, CRISPR technology is only about 10 years old now, but it's really revolutionized every facet of what we can do with biomedical and agricultural animals, and also is now really revolutionizing how we treat a variety of human health conditions, particularly blood-borne type diseases, with a lot of promise in terms of curing persistent diseases in humans. The importance of the pig is a really strong biomedical model and a platform to develop medical solutions. So all of these type of diseases right now, which are present in the human population, can be adequately modeled in pigs. And it's kind of fascinating, the biomedical and research applications that we can use with the pig as a model system. And a lot of this has come to the forefront with the recent xenotransplantation studies, where now human patients have received liver, a kidney, as well as hearts, and continue to survive with those xenotransplanted organs. And obviously, these individuals are not, they've been waiting on human organ transplant list and have been denied for various conditions. And so the pig really has a promise for solving this part of our healthcare system dilemma. The precision health efforts that we have at Mizzou include both biomedical to develop, produce, and distribute swine biomedical research models, and provide models and the expertise to tackle important diseases. Also, we have another dual program in terms of animal agriculture to answer basic biology questions important for agricultural research, to produce genetically modified animals to solve health and welfare issues, to ensure the security of our food production system. So we're currently producing animals that are resistant to a number of different viruses. There are other groups that are involved in food safety applications, such as bacterial resistance, biosynthesis of essential nutrients, improved feed efficiency for sustainability. I think gene editing in agricultural animals is going to be paramount to combat climate change, just like genetic engineering of plants has really helped us combat climate change. Obviously, there's a lot of animal welfare applications, and now currently through relatively high throughput systems, we can genetically engineer both pigs, but also chickens, which is an important food for particularly the U.S. as well as across the world. And so I'll just highlight at the end of the talk now a few applications. Well, here, this is actually a UC Davis offspring called Spotted, if I remember correctly. It's an animal that was genetically engineered, whose parents had horns, but now their offspring don't have horns. In addition, there have been, particularly in Europe, several groups that have genetically engineered birds to resist avian flu. We have right now an outbreak of avian influenza virus in the United States, and actually the highly pathogenic variant H5N1 has been recently shown in Texas to jump from cattle to humans that originally derived from wild birds. There's a lot of applications, particularly in the poultry industry, to be able to genetically engineer birds where you don't have the production of male offspring, where you only desire female offspring. So this is an animal welfare issue, so you don't produce an undesirable sex. And you can also genetically engineer different types of nutraceuticals as well as biologics that you can purify from eggs. And I think what is a really exciting development, there is a company called Genus that has almost final approval in the United States to produce a virus-resistant pig that will be able to be fed into the current food animal production chain. And that pig was originally made at the University of Missouri. So this is against a porcine reproductive and respiratory syndrome virus that causes pneumonia and diarrhea with a mortality rate in young pigs of about 12 to 15%. If the animals survive and go to adulthood, Many of them end up with severe reproductive failure, a high rate of miscarriage and premature birth with decreased litter size, and there's been no vaccinations that have been shown to be effective to date. So in North America alone, this costs about $600 million, about 1.5 million euros annually in Europe, and not including Asia, about $6 million each day. So this impacts the profitability of operations. It's an animal welfare issue. It's a food security issue. And there's also psychological and emotional costs to producers that have to basically depopulate their animals when they get these types of infections. And so I'm really excited about this first approval by the FDA for the consumption of these animals, which is probably a bellwether for the acquisition of more types of gene-edited animals that can be used to improve animal health and welfare. So I really appreciate the invitation to come. Hopefully, you learned a few things today through different types of things that I've discussed. And for finally, one of actually a former member of the National Academy of Science, a guy named Carl Hartman, who was the first person to get a PhD from the University of Texas, who was also a reproductive biologist, published a paper in 1967 that said research should spell fun. I like to define it as what you are doing when you don't know what you are doing. But it's been great, and so I'd be happy to entertain any questions that you have. And I look forward to giving another lecture tomorrow on a completely different topic. So thank you. You have to have the CL on both sides.
So there is no cross protection.
There is no cross protection. Not all species are like that, but particularly in cattle, you need both.
And my second question, just fascinated the development of the cotyledons in the coronal. And what's the evolution of placentation? There's so many kinds. Is this independent evolution, or is there any connection to these different plantations?
That is a really good question. I think is something, I see Harris smiling. It's something that I think all of us have, it's like a central question during the evolution of viviparity, right? Like, why do you get all these different types of not only mechanisms to establish pregnancy, but also phenomenal differences, like pigs, completely non-invasive implantation, to semi-invasive, to like humans and non-human primates, where you have amazing invasion all the way through the muscle of the placenta. And I don't really know particularly the penultimate answer to your question. There are cases where you have convergent evolution, where you have a fundamental event. So one of them is you have the acquisition of endogenous retroviral envelope elements that came from an exogenous virus that then were used to produce a unique cell type. So like the syncytial trophoblast, where you have a continuous one-cell layer of the primate or the human embryo, that's because of an endogenous retroviral envelope protein called syncytin. In many other species, you have the entry and propagation of those elements through the germline, but they always are of a different virus. Because most viruses, thank God, are not zoonotic, right? Or we probably wouldn't be here today. But I think maybe I realize I'm rambling a little bit on this question, but one thing is, I think, when you evolve one direction, and now you become dependent on that developmental process for the success and survivability of the species, it's really hard to go back, right? Because you probably won't make it. So it's almost like you have a fixed evolution toward these different types of placentation. They all have the same function where you're basically producing a fetus, but how they go about that is a lot different, and I find it fascinating. I still think it's still one of those things that we don't really have a full. That generally is true, yes. I mean, I think one thing, like, actually there was a famous anatomist here, Alan Enders, from UC Davis. You see a lot of the variation that you see in primates is actually amazing. I mean, you have some primates where you have almost no invasion, to where you have complete. Some of them have kind of a dual discoid placenta, even though, like in a human, you usually only have one disc. So it's pretty fascinating. I mean, it's like, menstruation, right? I mean, humans, some non-human primates, but not all non-human primates. And then for some reason, select species of bats and the spiny mouse. Like, yeah, very interesting. Thank you for those questions. Any other questions I can't answer? Yeah, I think those are excellent questions. One thing that we tried really hard was we used a single father, so a single source of sperm for every single embryo, and then we graded them like they were very high, at least, when you grade them microscopically, right, very appropriately developing and high grade embryos. So we tried to get around some of that. In terms of an experimental design issue. I think the other thing that at least we hoped was, because we consistently, we did this over and over and over, that we would basically be able to identify animals that consistently responded to an IVF embryo either one way or the other way. But it doesn't really solve kind of some of the experimental design questions. And that's correct. And I think, like, if we were going to redo it, which probably, hopefully, we will never do again, it just logistically is very difficult. I think if you could take, and you know, since you got two horns, if you ligated one off and then you created a pregnancy by natural insemination, and then the other horn you put in an IVF produced embryo, maybe you could get around that. I think the other area that, and I know, I remember the work that you did as well as another group from Germany. I think, too, it would just be doing a large enough number of animals where you got enough observations to statistically and bioinformatically segregate the responses out. But I think, like, maybe on the dual purpose efforts, when, you know, we got NIH level funding, you could kind of do that. But with today's USDA grants, you would never have enough money to do it. Yeah. You know, that's a good, very good question. I mean, essentially, you know, IVF, at least in the United States, is really not regulated. I mean, you know, and even reporting pregnancy rates and outcomes to the Centers for Disease Control is actually voluntary. It's a voluntary process. It does impact the ability of those clinics to recruit patients. That's why most of the clinics do it. You know, I think at the end of the day, like, we know the embryos aren't normal. There's been a lot of refinement with, you know, now, you know, biopsies and looking at pre-implantation genetic diagnosis. That has improved the pregnancy outcomes in certain circumstances. But I think one of the problems, at least in the U.S., is we haven't had enough IVF programs that have created enough babies and had the potential to follow those up. I think now with some consolidation in the programs, like if you look at, you know, on the East Coast, like RMNJ in New Jersey, Shady Grove that comes out of Bethesda, like, you have enough observations where you might be able to start follow-up studies. One of the problems is we have such a fragmented healthcare system that really doing longitudinal follow-ups is almost impossible. And most of those studies have, at least in the UK, where you have a more socialized healthcare system that pays for this, there's been relatively little follow-up in terms of the epidemiology. And I think it's an important problem. It will be very interesting with, I would say, the political dynamics of the United States and the fetal and embryo personhood movement about, my prediction personally, being from a state that has more conservative politics, you're going to see a real reduction in the number of babies that are born to IVF. Right. Yeah, or maybe some of the new microfluidic approaches where you can basically sample what the embryo is secreting. That one might be where you really aren't having to do like a cell biopsy approach. I think that would be very interesting. I think the other, the other area, to be honest, and not to belabor the conversation, is when you start messing with human embryos, you get into problems with actually obtaining funding by the National Institutes of Health. And that can become problematic. And I think that that's one of the things that, and particularly which state you're in, can also become problematic. Right. And a lot of these outfits that are doing this, they just don't have a research budget, right? I mean, or they have a small research budget, they just can't afford it. But I think, particularly when you're like, California, that would, without a doubt, that would be a great project that you can do because of the way that the state funds things, and also, that's right. Yep. Yep, I agree. You're welcome. Yes, ma'am. Sure. Wow, that's a good question. I like that question. I think why it evolved in certain animals, I don't really, let's say, like, why do certain bats menstruate? The weird spiny mouse, that's an interesting one. I think in humans, really, so what we found out over the last few years is that during a normal cycle, a normal menstrual cycle, that a lot of the epithelial cells in the upper part of the endometrium actually have cancer driver mutations. And so this is some work predominantly done out of Johns Hopkins. It's beautiful work done by Jim Seegers and Yiming Shi. So naturally, right, if you have these mutations that we know are cancer oncogene drivers, then shedding that out before they actually become cancerous is a good thing. And so what supports that hypothesis, right? I mean, most women that develop endometrial cancer, this is a cancer of women that are going through menopause or have gone through menopause. Very few, reproductive age women, I would say, like between 15 and 40, very few rates of endometrial cancer. But when you stop shedding the endometrium every cycle, and now those cells can take over the ones that have the cancer driver mutations, that's when you develop mostly benign adenocarcinoma events. So I think that's that in a large part allows for that. So, yeah, thanks for a great question.
Following that up, then how do you explain the one in the carotid chronicle system where there is really huge separation, right, between the mother and the thing? Is there any chance for cancer to develop in that system?
It turns out it's kind of amazing that most domestic animals never get cancer. And only, it's kind of like Hereford cattle that get, they don't have pigment in their eye, so they, have predisposition to basically eyelid type cancer. But most animals, and it's, to be honest, I think it's not because, to be honest, most animals have a defined lifespan because then we eat them, or they end up in the food supply. Even if you age cattle, like people that keep, there are beef heifer, like cattle that are walking around that are, 10 to 15 years old, no problem. And it's, I think, amazing when you think about every breed went through a genetic restriction point to develop that breed, right? Whereas if you look at dogs, it's like the reverse, right? I mean, my beagle has epilepsy, very common in beagles because, the genetic bottleneck predisposed those animals. You have many different canine type cancers that are prevalent in one versus another breed, but yet in domestic animals, they just really never appreciably develop cancer, which I think is amazing. Even horses are amazingly resilient. Good question. Stuart.
Thomas, can I ask you, from your position as an institutional perspective, what's going to happen to the xenotransplantation world? Is it going to explode?
Personally, I think it is going to explode. And the reason I think that, number one is you have a couple of different major companies involved in now, not just one. It's not just Revivicor. You have iGenus and a number of other companies that have made their own lines of immunodeficient pigs that you can use for xenotransplantation or lack, the antigens that cause transplant rejection. I think the other thing is just because as we live longer as a population, particularly in the United States, we just have a huge number of people that are on organ transplant lists that desperately need organs, and we do not have enough donors. Because I think that's one of the consequences of actually improving our lifespan is that there's naturally less organs available, and most of these are most of the organ donors are derived by accidents. People unfortunately have their demise, and that nationally is a lower trend, and so I think it's going to explode. And some of the, you think now it was originally just heart, now it's liver and kidney, and I think it's going to be an amazing biomedical application of it. Yeah, I think it's, and the FDA is already approving these procedures, and it's not, fly-by-night medical schools that are doing these transplants. It's Penn, Philadelphia. They've been ones in California now, and so it's I think going to be rapidly available to improve the human condition. Thank you for that. All right, thank y'all. Oh.
Sorry, I'm a virologist, but I find this really fascinating. My last question. Are there any differences? How likely is different transplantation systems? Has anybody done a comparative study how it protects the fetus from being rejected?
Yeah, I think it's a good question. Most placenta, lack classical class 1 MHC unless something goes wrong during placentation. And so now they're recognized. And then really the endometrium, particularly like in humans, right, the specialized decidual cells, they are responsible for kind of keeping the maternal immune system at bay. So, and I think personally Like in cattle, I don't think the mother, from an immunological perspective, actually ever recognizes the actual conceptus allograft in terms of the placenta. But it's very reduced compared to. Yeah. Right, but we could talk about horses. Yeah. That's amazing. So, well, thank you all very much for your attendance, and I appreciate your time.